Hi everyone, thank you all for coming. My name is Zachary. I'm from Louisiana State University. I'm working with Dr. Christopher Austin at the Museum of Natural Science. Thanks. Today I'm very excited to talk to you about a complex trait found in Guinea lizards and how I use phylogenomics to investigate the evolutionary history of this trait. First, I want to give you a little introduction to my study system. Presention is a genus of arboreal tree skinks endemic to New Guinea and surrounding archipelagos. They have a broad range and occupy a variety of different habitats, from montane primary and secondary forests at elevations higher than 3,000 meters, all the way down to lowland forests and coastal forests. Like other Australasian lizards, they also exhibit a variety of extraordinary morphological and physiological modifications. For example, some have adhesive toe pads, many give birth to live young, and a couple even have prehensile tails. But one extraordinary physiological change has occurred in this genus at the exclusion of every other amnio, and that is the evolution of green blood. Now, the green blood actually diagnoses this genus and gives the genus its name Presention, which literally translates to green blood. Now, the green coloration actually extends to the blood, muscles, bones, and even the mucosal lining, as you can see in this awesome shot of the Presention prehensicata. Now, the green coloration is actually due to elevated levels of the biopigment biliverdin, and this overwhelms the normal levels of the crimson ferrous form of heme that you would see. If you've ever had a bruise, biliverdin is responsible for the greenish blue tint that you see. And this is because biliverdin is a waste product of red blood cell catabolism, and they're essentially modified dead red blood cells. Now, normally in other amniotes, biliverdin is rapidly filtered out of the bloodstream and quickly disposed of through the liver. This is because biliverdin is cytotoxic, neurotoxic, and causes a pathological condition known as jaundice. Humans keep a biliverdin concentration at or below 50 micromoles per liter. Yet these lizards exhibit a biliverdin concentration between 250 and 700 micromoles per liter. At this kind of concentration in other amniotes, biliverdin would diffuse out of the bloodstream, cause cell membrane lysis, and even general DNA damage to the production of reactive oxygen species. This is particularly worrisome in newborns, whose livers haven't fully kicked in yet, because biliverdin could actually pass through the blood-brain barrier and cause permanent nerve damage. Now, this begs the question of how on earth are these lizards surviving in this cytotoxic environment, and why do they even have green blood? We have several hypotheses regarding this. Green blood may confer antioxidant or cytoprotective properties. It may provide cryptic coloration, as seen in this prehensicata here. There may be some thermoregulatory advantages, such as the protection against UV radiation. And the toxicity may even deter infection by Plasmodium parasites, which are the causative agent of malaria. Now, my advisor, Dr. Austin, is working with Susan Perkins from the AMNH to investigate this possibility. But before we can make any evolutionary inferences about green blood or the evolutionary origin of green blood, we first need an accurate phylogeny. Unfortunately, phylogenetic reconstruction for this group has been historically difficult. Early molecular phylogenies have short basal branches, which suggests there was an early rapid diversification event. Now, we hope to alleviate this with extensive genomic and taxonomic sampling. Luckily, I started my PhD in the age of phylogenomics, so I turned to the sequence capture of ultra-conserved elements, or UCEs. And this has been developed in part by the researchers at LSU. For this project, I targeted over about 5,000 UC loci and included 96 green-blooded and red-blooded individuals. This encompassed 56 species, including many undescribed taxa, such as this Presention here. I did the library prep and sequence capture myself and sent it off to Oklahoma for next-generation sequencing. After I got the data back, I cleaned, filtered, and aligned it, and I created several different data sets. I had an all data set, a 1K dataset or 1,000 dataset, which included all individuals with more than 1,000 UC loci, and I also had a main dataset, which is just a pared down dataset for even taxonomic sampling. I also examined the effects of missing data by allowing my alignments to have varying levels of matrix completeness. I also examined the different levels of information content by sorting UCEs by number of parsimonious informative sites and looking at those different levels. I ran all of my datasets and subdatasets through common phylogenomic methods such as concatenation, coalescence, and species tree approaches, using many common methods such as the ones listed here. Overall, I recovered a well-supported species tree with topological concordance across datasets. Now, I have many, many trees, but I have one well-supported result that I'd like to share with you. And that is that Presention is not monophyletic. Now, this isn't as surprising as you would think. The single character that diagnoses this genus is simply the fact that they have green blood. And there's a lot of, as you can see, there's a lot of interspecific morphological variation. Now, the next thing I want to investigate, because of this surprising result, is how many origins of green blood are there? And are there any reversals back to red blood? Now, despite accurate phylogenetic reconstruction, I have several hypotheses, 3 of which I want to share with you today. Now, before I go on to my next slide, I've been told many times not to use green and red in presentations, but I'm going to for scientific clarity, and I want to apologize for anyone who's colorblind in the audience. The first hypothesis is a multiple origins hypothesis. This is best explained by 4 independent origins of green blood. One such origin is the sister pair right here, and it's morphological similarities. The next hypothesis is also a multiple origins hypothesis. This is best explained by 3 independent origins of green blood, followed by 1 reversal back to red blood, and you can think this is the pineal longiceps here. You can also notice the morphological similarities between this sister pair. The last hypothesis that I want to share with you today is a single origin hypothesis. This is best explained by a single, more ancestral origin of green blood, followed by multiple reversals to red blood. Now, with 5 total changes on the tree, this tree is actually less parsimonious than the first 2 that I presented. But we're not going to rule it out just yet. Green blood is a seemingly difficult trait to gain because it's not present in any other amniotes. And every time green blood evolved, it would have had to develop some sort of coping mechanism to deal with this cytotoxicity. However, if these lizards are experiencing similar extrinsic selective pressures and have some, they may be predisposed to green blood if they have some shared underlying developmental or genomic architecture. On the other hand, if there aren't any major physiological restraints on the reversals back to red blood and extrinsic condition promotes that reversal, then a single origin could be likely. So in summary, I've provided a well-supported species tree, and this has laid down the evolutionary framework not only for my dissertation, but for future work on the system. The next obvious step is to figure out why these lizards have green blood. For that, I'm currently sequencing the transcriptomes of lizards with red blood and sequencing the transcriptomes of lizards with green blood and closely related lizards with red blood. And I hope to get to examine if there's any genomic or proteomic innovations associated with the evolution of green blood, or how they might be able to deal with this cytotoxic environment. Only then can we start to get at some of these adaptive hypotheses. There's many people I'd like to thank for this project. Of course, my advisor, Dr. Austin, everyone in the Austin lab, collaborators at LSU and beyond, the funding agencies, NSF, including the Graduate Research Fellowship, and of course the natural history collections, which I received many tissues for this project. Again, my name is Zachary. I'm from LSU. My contact information is up here. I'd love to hear your thoughts, comments, or questions about the ideas I presented today. And with that, I'd be happy to take any questions. Thank you. Yes?
Do you or anyone have any speculations as to what the advantage of having green blood or being full of billiebird might be?
Not yet. We do have these hypotheses that I presented here today, such as cryptic coloration, but I don't know, it's hard to say now. Again, my professor and Susan Perkins are both looking at the possibility of an anti-parasitic strategy to determine malaria, but I'm more interested in how these lizards are even surviving in this cytotoxic environment. And that'll come soon, maybe at evolution.
They're not poisonous, are they? Like if an animal ate them?
Nope. Dr. Austin has conducted feeding experiments and they're readily taken.
What are they
Yes.
Are the green blood species restricted to high elevation?
No, and that's the interesting thing. They're all arboreal, but they're found at high elevation, mid elevation, low elevation. Some have adhesive topaz, some don't. Some give birth to lions, some don't. So hard to find what's going on. Any other questions?
I have one more question.
Sure.
So Presention, I thought that was the third example of a lizard group that has expanded topaz with microscopic seeding. Is that not monophyletic also? Is that trait not one origin? So these things have green blood and the topaz? Convergent label is
Well,
potentially.
Sorry. Yes. More future work will figure out that. But there's multiple topaz morphologies, which is pretty cool. Yes.
I may have missed, but across that filter, did you find difference in the topology of the trees?
I'm sorry?
Across the filters that you use, 10%, 30% PC data, and also the low side, did you find any difference?
No, there was topological concordance across A set for the most part. They're all supported. I was happy. When I first saw the non-mono file, let me read a bunch of more data sets, but they all gave me the same result. I even looked at, I conducted a lot of monophyly tests, like SH tests, and seeing how many trees, if there's any trees at all that support monophyly of Presention. I think there was one UCE gene tree out of 3,000 that did that. It was very poised. Well, thank you all for coming. I'll be here. Do you have any other questions?